Now, this concept explores how different solutions impact plant cells, focusing on turgor pressure and plasmolysis. The diagram on the board is divided into 3 sections, each depicting a plant cell in a different solution to illustrate these processes. On the left, in a hypertonic solution, the plant cell is shown as turgid, with water molecules entering the cell, maintaining its shape and pressure. In the middle, in an isotonic solution, the cell is flaccid, with water moving equally in and out of the cell, leading to no change in size or pressure. On the right, in a hypertonic solution, the plant cell is plasmolyzed, with water moving out of the cell, causing the cell membrane to pull away from the cell wall. Each plant cell in the diagram is shown with a thick outer cell wall and a thinner inner cell membrane, clearly illustrating how water potential affects the position of the membrane and the cell's turgor pressure. This image effectively demonstrates how plant cells respond to different water environments and the role of osmosis in maintaining cell integrity and overall plant health. A turgid cell is one that is swollen or distended, typically due to high fluid content. Turgor pressure, also called turgidity, is the force that pushes the cell membrane against the cell wall, which is caused by osmotic flow of water into the vacuole. Healthy plant cells are turgid, as plants rely on this state to maintain rigidity and structure. In a turgid cell, the plasma membrane is pressed tightly against the cell wall. This condition occurs in a hypertonic solution, where water enters the cell and causes it to become turgid. The diagram on the whiteboard shows a plant cell in a hypertonic solution, with the cell membrane pushed against the cell wall, labeled as a turgid cell. A flaccid cell is one where the plasma membrane does not press tightly against the cell wall. In this state, the concentration of water and solutes is the same both inside and outside the cell, meaning there is no net movement of water. As a result, water molecules move equally in both directions, in and out of the cell. This condition occurs when the cell is placed in an isotonic solution. The diagram on the whiteboard illustrates a flaccid cell, where the cell membrane is slightly detached from the cell wall in some areas. A plasmolyzed cell is one in which a significant amount of water has been lost through osmosis, causing the plasma membrane to detach from the cell wall. This happens when the surrounding solution has a lower water potential, higher solute concentration, than the cell's internal environment, resulting in water moving out of the cell. A cell becomes plasmolyzed when placed in a hypertonic solution. The diagram on the whiteboard shows a plasmolyzed cell, where the cell membrane has completely detached and shrunk away from the cell wall. Plant texture is maintained through a balance between the cell wall and the vacuole. The vacuole, which stores water, pushes outward on the cell wall, helping to keep the plant firm and rigid. Meanwhile, the cell wall provides enough mechanical strength to contain the vacuole, preventing the cell from overexpanding. The diagram on the whiteboard shows 2 plants. The one on the left is upright and healthy, with a turgid cell, while the plant on the right is drooping and wilted, with a flaccid cell. This illustrates how proper turgor pressure is essential for maintaining plant texture.